Welcome everyone. Today's public webinar series. As a reminder, the public webinar series is designed to educate attendees on the scientific ecosystem, and each month we'll have representation from a data coordinating center or program within the CFDE to show new developments from all of the groups that support the CFDE. So with that today, I'm going to go over a few KC updates, share some upcoming events, and then I'll hand it over to our featured speaker. Again, if you're unable to attend last month's webinar, the webinar recording is available on our YouTube page. This session will be recorded and will be uploaded as well if you're interested in sharing with your colleagues. If you'd like to give any feedback, questions, or you'd like to be added to our listserv, you can email us at help@cfdeknowledge.org. As I mentioned, we have a few updates for events. So this past week on Tuesday and Wednesday, we were able to attend the Festival of Genomics and Biodata in Boston. This is the second year that we have had the CFDE at this event. This event was absolutely fantastic. I just want to thank every single person who helped make this a success. We had both a booth and a Genome Dome talk, and this event was so well attended. We had about 300 people between the 2 days stop by, ask scientific questions, and eager to learn more about CFDE. So we are super excited that this, again, is just another way for us to educate people on CFDE and some of the programs that are involved. So thank you again to everyone for making this such a success. We look forward to being at this again next year, and it's going to be in June again, so keep an eye out. And again, thank you to everyone who made this such a success. And with that, I'd like to share some upcoming events that we have in terms of training and outreach. So upcoming in July, we will be attending the Intelligent Systems for Molecular Biology in Liverpool. And so there are a bunch of different CFDE programs that are going to be attending this event, but I'd like to highlight the Knowledge Center's Julie Juergens, who will be presenting the GeneSet browser, an interoperable and AI ML-ready tool for gene set analysis in the Common Fund data ecosystem. If you're going to be attending, this will be part of session 2, Cyber Infrastructure and Emerging Technologies, on July 21st. So if you're going to be attending, please keep an eye out for that and for all of the other talks that are going to be ongoing through this session that feature the CFDE. I know there's a bunch of different groups and programs, so I just wanted to highlight that if you'll be attending. And as we did last year, we are going to be attending the American Society of Human Genetics, which will be in October in Boston. The CFDE will be having a booth, an ancillary session, and a networking event. So we're starting to talk and publicize about this now, and I know it doesn't seem far away, but it actually isn't. We're really excited about this. We're hoping that CFDE community members, even if you're not attending ASHG, but you're in the local Boston area or New England area, we'd love to see you at these events as well. We're hoping to get a bunch of different groups there to all share the science and educate attendees on all the hard work that we have in the ecosystem. So if you're going to be attending or if you're interested in learning more about these events, please feel free to reach out to me or our help email address, as we'll have numerous events. And as we get closer to, I will be sharing all the details for this as well. One other thing that I'd like to highlight is the Decoding the Data Ecosystem, a CFDE Training Center podcast. And this is hosted by Dr. Alyssa Dillman from the Training Center. And this is more than just a podcast. It's actually a community for anyone who's passionate about the potential omics research to solve complex biological puzzles. And it's also used to address pressing health challenges. So this is for anyone, whether you're a seasoned researcher, you're just starting out as a student, or you're just curious about biology. So this podcast offers insights, inspiring stories, and practical advice to guide your journey through the role of omics research, training, and education. And so with that, those are all the updates that I have in terms of our events and from the KC. So now I'll be handing it over to Andy, who is going to be presenting Multiscale Virtual Reality Exploration of the 3D Human Reference Atlas provides novel insights into health, aging, and disease. So with that, I'll be handing it over to Andy. Thank you so much for joining us today.
Thank you so much, Mackenzie, for the kind invite. I did put some links into the chat about things that I'm going to talk about, and you should now be seeing my screen.
Yes, that looks great.
Wonderful. Thanks, everyone. All right. It's an honor to be here today. I want to report about my ongoing HuBMAP JumpStart Fellowship about the usage of virtual reality for exploring the 3D Human Reference Atlas. Again, my name is Andy. I'm the research lead here at the CNS team at Indiana University. So there is a long-standing vision for the usage of immersive technologies such as virtual reality, augmented reality, or mixed reality for the usage in medicine. And oftentimes the ideas take on rather sci-fi dimensions. What you can see here is obviously a stock photo between two people, nurses or surgeons, working in an operating room with this kind of holographically displayed brain floating in front of them. Of course, this is complete science fiction. None of this currently exists. And if you look closer, you will see things like, well, this one is not wearing a headset. So is this just like a Star Wars-like apparition in the middle? How is this possible? And even if they did that, what exactly would they be looking at here? Is this an actual representation of the patient's brain here? We don't really see any data overlay either. So I did a little more searching. There's additional, I think that's probably from the same photo shoot. And this time they actually gave them headsets. These actually look pretty useful. I don't think anything of that nice form factor currently exists. It kind of looks like a shrunk down Apple Vision Pro, but my assumption is it's probably just a prop that they used for this particular shoot. And then you have other images. Those always look really cool when you use them for promotional materials. But if you look a bit closer, you will notice that it might be rather hard to use in an actual setting. Well, one thing that I stumbled upon here was that these are all different headsets. Usually, if you have these kind of group sessions, everybody's wearing the same type of headset. And then this person here, for one reason or another, cannot actually strap the headset to their head, so they have to hold it with one hand, and then that of course makes collaborative exploring here a lot harder. So there is a long-standing vision for using of these technologies, but what's actually practically possible today is quite a bit different. The reality looks more like this. A lot of virtual reality and augmented reality applications are used for learning anatomy and physiology. That's a natural use case for it. In VR and AR, you don't have to worry about pesky things like gravity when you, for example, want to dissect a human body. You don't have to worry about it decaying in front of your eyes either. And so these are two examples that I pulled from existing learning applications. This one is for the Meta Quest, which is the platform that we also support. And this one is an application that comes in an AR and a VR version. And so the models that are used to they vary in detail from photorealistic to more like low level of detail so that they run more smoothly in the lower grade hardware that is supported here. And none of the existing applications, as far as we could tell, though, display patient data and definitely not in large numbers. And the concept of what we're trying to build here based on these gaps is a so-called information-rich virtual environment. It's a rather old term. It was coined in 2003 by Bauman and colleagues, where they defined an information-rich virtual environment as a realistic virtual environment, so a 3D world that is enhanced with the addition of related abstract information, so information that is not inherently spatial. And the examples from that paper, again, this is 2003, what you see here is a human throat. We have in the foreground the tongue. We have some vasculature here in the back. And then we have, and there's a zoom in here, a palatine tonsil. And then this is some of this related abstract information. So in this case, it's just the surface area of this particular 3D object. And they also bring forth the idea of these semantic objects. So these are 3D objects that live in 3D space, but they have associated 2D information. And of course, nowadays, with the proliferation of video games on basically everybody's smartphone, we're a bit more familiar with this concept. But at the time, it was really forward thinking. And so I'm going to put a pin in this for now. I'm going to get back to this concept later. So I want to talk about the exploration of the Human Reference Atlas in virtual reality. So I want to briefly talk about the Human Reference Atlas. So the Human Reference Atlas is a comprehensive high-res and 3D atlas of all the cells in the healthy human body. HuBMAP and ZenNet, which we are funded by, partially contribute to the building of the Human Reference Atlas. This is a cover of Nature Cell Biology that we made a couple of years ago now. So it is funded by a series of consortia, and it defines the 3D space and shape of anatomical structures and the cell types that are found inside of these anatomical structures, as well as the biomarkers that are used to characterize these cell types. And all of these anatomical structure cell types and biomarkers are validated and also represented in related ontologies such as Uberon, FMA, cell ontology, or HDNC. So we love ontologies here. Now, the Human Reference Atlas also defines how new data sets can be mapped into it. Usually that is done spatially, and that is mostly what I'm going to be focusing on today. The Human Reference Atlas provides linked open data, for example, positional data, so spatial, biological structure, or specimen, that's donor data. And the HRA itself is authoritative in that every single unit of knowledge captured in it is made with with expert agreement, and it was validated by scientific evidence where available. It is computable in that it supports API queries and UIs, and is also accessible via a large-scale knowledge graph. It is published as linked open data, so every unit of knowledge is ideally linked to an ontology term or an ontology relationship. It is open in that anyone can use the HRA data and code or also contribute to it. And it is continuously evolving, for example, with new technologies becoming available with releases twice a year, every 6 months. We just had our last release a couple of weeks ago on June 15th. Another thing that we put forth recently is a Nature Methods resource that describes the HRA construction and usage. The paper is here. I also embedded this QR code here so that you may peruse the paper. This paper had over 170 authors, so as you can imagine, it's a major contribution that we were trying to make. In my talk today, I'm going to focus this is from Figure 1 of the paper. I'm going to focus particularly on the 3D aspects of the HRA. Before I do that, a bit more about this paper. This is what it looks like. Another thing that we supply are high resolution supporting figures. There's, if I remember right, 17 of those. We have a series of supporting tables that link to all of the data and code that are used to construct and use the HRA. A link to our HuBMAP paper publication page, and then also the GitHub repository that holds some of the additional code that is used for it. So again, 170 plus authors. We had 19 core authors and then literally hundreds of HRA team authors across a total of 26 consortia. Another core aspect of this paper are the HRA user stories that come out of 30 plus, at this point I think 50 or 60 plus, one-on-one interviews that one of our postdocs, Dan Koruni, did with various Atlas architects and programmers and developers and researchers across all of these consortia. And we isolated 7 user stories that came out of this. And then we did have quite a bit of nice press coverage, some shout outs on Twitter. We had some press here locally at Indiana University, and I compiled all of this here as well. So again, the HRA is a gigantic beast. What I'm focusing on today is a small slice, and that's mostly the 3D part. And here we're going to focus in on VR. You can see that again highlighted here. So major components of the Human Reference Atlas version 2.3, which is the most recent version, are 73 anatomically correct 3D reference objects for 33 organs. We're going to look at those in detail, but you can see, for example, the male left kidney over here. And here we can see a variety of organs, including the male and female skin. You can also see them represented down here. 34 anatomical structure, cell types, and biomarker tables, or ASD plus B tables. And soon also we're going to feature cell type populations in 3D. This is particularly important here for CFDE. That is some work that we're going to accomplish over the next couple of months. And I want to focus on a few HRA applications here. So HRA data is available programmatically via APIs, but also via user interfaces, of which the organ gallery, the VR part, is one. So there is the registration user interface, the exploration user interface, that's those two. We're going to look at those in a minute. The AST plus B reporter, and then also the HRA organ gallery and virtual reality. So let's have a closer look at these user interfaces. This is the registration user interface. It's a web deployed interface that is embedded in the HuBMAP and ZenNet data ingest portal, but it's also available as a standalone user interface. It allows the user to pick one organ. In this case, this is the kidney. And then here we only see the renal pyramids inside of the kidney. And then the user can move this yellow tissue block into the extraction site when they want to register a sample. So this allows the user to indicate from where inside of this reference kidney a tissue block was extracted. And then the system interface to the RUI is the EUI, or the exploration user interface. Here the user can see every single tissue block that anyone ever has registered into any of these organs. And so again, there are 73 of them. And here, particularly, we see a zoom into the lung. So this is the female lung. We can also see the liver down here, and then there's the heart embedded in here as well. And again, there's 73 organs. We don't see all of them here. I only turn on the ones that have turn off registrations here. And so this kind of over time creates this visualization of which part of the body has been mapped to what degree. And so there is a tie between the 3D reference objects and all of these anatomical structure data. So here we see a close-up of this lung, the male and female one. And here we see bounding boxes. Those are all-encompassing cubes around every single anatomical structure that is modeled inside of that reference organ. These models have been custom built by our medical illustrator team with input, of course, from subject matter experts. Nobody in any team can know it all. So we bring in experts for all of these different organs that review and validate these organs with us and for us. And these organs are then used in the RUI, EUI, and also in VR and the H-ray organ gallery. So now let's talk about the H-ray organ gallery. I mentioned earlier that we have the exploration user interface, which is a 2D interface that runs in a web browser. So we were asking, what if? What if instead of enabling the user to only view these organs on a fixed two-dimensional screen with the old established input devices of keyboard and mouse, what if instead we place the user right next to these organs at their true scale? All of these models have been made, most of them, with visible human data by the NLM, and they are all modeled at real physical scale. So this allows the user to explore these organs and also make them bigger and smaller in our own spatial reference system that is our body, which we have with us all the time. So here's a link. We built the application and we started deploying it via the MetaQuest store. That is, we have found that is the easiest way to distribute software for these MetaQuest headsets. And we also wrote a paper about it. This is our first paper. We're currently working on a follow-up one. It's old, it's from 2023, but it explains the basic idea. And then here again, you can download the application to your own headset. This is what the Meta Horizon Store entry looks like. If you use a platform like Steam or GOG or Epic Game Store, it's the same thing, except it's for VR. And so we have an entry there that allows anyone to get this application with one click of a button, essentially. So in this recent JumpStart fellowship, the idea was to add more multi-scale exploration of spatial data into the organ gallery. So initially, all the organ gallery supported was the exploration at the whole body scale. But as you all know, as biologists and people who work with biological data, the really interesting thing comes at different levels. Or once you start traveling across different levels into a multiscale environment, to organs, to functional tissue units like the renal corpuscle, down to cells, and then ultimately down to genes. And the inspiring concept and framework for this work was the Powers of 10 movie. It's a 1977 movie that many of you might have seen. It starts with a couple that has a picnic here at Burnham Harbor in Chicago. And then the movie starts increasing the point, like the shown view by one order of magnitude, one power of 10 every 10 seconds. And what starts as a relatively slow zoom, where after about 40 seconds, you can see, you can see the harbor here, you can see the old Burnham Airport over here. But then after a couple more minutes, you suddenly at the edge of the observable universe, because we move in powers of 10, and that's not something that we are used to as humans. We're used to more linear scaling. So it's a really educational moment. And then the movie goes back in towards the couple, and then it starts going into the man's hand. And here as well, as we are traversing these individual scales, we start seeing smaller and smaller things that at some point we cannot see with our eyes anymore. So at some point we break through the skin, and then at some point we're down to the DNA helixes, and then at some point we're down at the atomic level. And of course, we get quickly to the boundaries there of what they were able to visualize at the time, or what we could even know at the time. So we took all of these inspirations from this Powers of 10 movie, and we organized a Powers of 10 workshop that my research assistant Natisha helped me with here on the top right. This was at the BioVis lab, at the Biocomputational Bioinformatics lab at NIAD. We did that in October last year. So this is the building. They have a beautiful biovisualization lab in there, which for a VR developer like myself is basically heaven because there's a lot of walkable space, so you don't run into walls all the time. And they also have all of the VR equipment that you could dream of. So for us, this was an extremely valuable way of introducing, of like trying out different hardware, but also introducing other researchers that don't typically work with VR into this. So I was able to invite four excellent other early stage investigators from Harvard, Duke, Yale, and Stanford to join me for this workshop at NIAD. So none of them have VR experience, but they all have data. So they brought their data and I brought the VR experience. And then together we had an amazing 2 days of hacking and prototyping and analyzing. And what we were also able to do was bring in NIH NIAD personnel. So these were some amazing people that we have worked with for the past couple of years for hosting these kinds of hackathons to bring in experts and discuss and hack VR in person because that is really easier to do if you sit next to each other and you can try out these headsets together. So this was a fantastic experience, and I'm really glad for everybody who could join. Another nice perk of doing this in person in a lab like this is that you can try out all of these different VR headsets. This is Chen Chen here wearing a Quest Pro, and this is all of us here wearing a variety of headsets. This is a Quest 2, this is a Quest 3. We had an Apple Vision Pro there too. And some people work better with different headsets, and so having all of this hardware together was a really, really important thing to have. And so for the organ gallery, we envision the human body as an inverted skyscraper, where you start at the highest floor, up here at the whole body level, and then you can take an elevator across different levels as you're being shrunk down, and you're able to explore increasingly small scale data. So the challenge then here is to map in data from collaborators into the correct level in our elevator. And so there is of course much construction going on. So that's why you can see all of these construction symbols everywhere. But there are a few levels that are currently open for business. So again, we have a whole body level. We're going to go through all of this in a second. We have a small organs level with data from Archie Enninful from Yale University, who is in ZenNet, who some of you might know. We have a large FTUs level with Hemali Fadnani's data here on the 10 to the minus third. And then we have the very popular cell groups level, where we have data from Clarence Yap from Harvard Medical School and Yang Miao from Duke University out of John Hickey's lab. And then we have an organelles level here at 10 to the minus 7 with Chloe Lindbergh's data, she's a PhD student at Brown University in Nicola Nareddi's lab. And then there's a variety of other levels that we're currently planning and prototyping with. And so one challenge that we early on did was to think through, well, how can we align all of these scales with each other and how can we communicate to the user when they are in virtual reality, what scale they're currently at? And so each level has a power, like how every floor in an elevator or in a building has a number, each level here has a power. And that indicates how much smaller a 1 meter cube would be. And so this is how we then try to align all of these levels with each other. And so you can see, for example, here we took screenshots from the Powers of 10 movie, and then we tried to align all of these numbers with each other. And another point of inspiration here that was originally the inspiration for the Powers of 10 movie is the book Cosmic View. This is a book by Kees Bueckle, who is a Dutch illustrator who made these beautiful drawings in the 1950s of things at different scales. It's the same idea as Powers of 10, except it's a book and it's static. And then my research assistant Juhi worked with me on making these beautiful multi-scale skyboxes. One of the advantages of being in VR is that you have a full 360-degree environment, and in video games, which is what we use a video game engine to make the application. This allows you to place a skybox. It basically, you can add clouds or the sun. We, in our case, add an object into the sky that is 1 order of magnitude larger. So for example, if you're at this particular level, which is the small organs level, then the next order of magnitude is a tennis ball. Or if you are 1 level down at the large FTU level, the next biggest item is a ladybug. And then below that, the next biggest item is a snow crystal, etc. So this way, it's another way to communicate scale while the user is situated in VR. So with that, I want to actually take you on an elevator trip through the human body And of course, that's always a bit challenging to do on Zoom because it's really better experience in VR, but we're going to make do with GIFs and screenshots. All right. So let's look at the whole body first. Here's an illustration of what that looks like in VR. So we have the male and female body suspended here up in the air. We have a user that is currently exploring the heart. And we can also see that we have a keyboard here that allows the user to select these individual organs. And this is what the interaction looks like. The user can make this organ bigger and smaller by using their hands. It's kind of like zooming into a photo. You move your hands apart and that makes the organ bigger. And that also allows you to move and rotate it as you go along. This is already an interaction that is very different. It works very different on a 2D screen. And with hands, it's natural and you can make the heart basically unlimitedly large and dive into it and see the nooks and crannies and the anatomical structures. But of course, that's all relegated to the whole body level. So then another level that's currently under construction is this level here that we built together with Archie for small organs. So this shows the human lymph node, about 40,000 cells. And as you can see, these cells here are colored by individual colors. And then as the user hovers over the colors here, the corresponding cell type gets highlighted here as well. And then let's go down another level to the large FGUs level. Now we are at the point where 1 meter actually corresponds to 1 millimeter. And so this is a Visium slide. I assume most of you are familiar with Visium. It is laid out in 3D on the ground, kind of like a carpet. And then we can see the individual Visium cell spots also laid out here. It's about 5,000 of them. And what you see coming out of the ground here, what kind of looks like tall grass, those are little bars that visualize biomarker expression values. In this case, particularly senescence hallmarks. And there's 8 of them. And you can see that they're color coded. There's a legend here in the background. And so this allows the user to understand, is there a spatial pattern to particular biomarker expressions? And also, are there differences between biomarker expressions for the same cell spot? Each of these cell spots here has 8 of these bars coming out of the ground. There is a video for this where we paired with Hemali Fadnani here from Columbia, she's from Columbia University, and I am in VR going through and demoing stuff, and Hemali is talking over it and narrating what we're actually seeing, kind of like a nature documentary. And so as we move forward with building out all of these scenes and floors with all of these experts, we're building similar videos so that people without VR headsets can experience these scenes as well. Now we go down another level to one of our oldest scenes. We built that one last year together with Clarence Yap from Harvard Medical School. This one is from the human skin. This is a melanoma data. In this case, we see about 11,000 cells in 3D, 13 cell types. And you can see the data laid out over here. And you can see that we have a legend on the left that pairs a color with a cell type and then the number of instances for that particular cell type. And here's an animated version. The user can do a dual hand zoom at 65 to 72 frames per second. That's a high frame per second rate. We need that to prevent motion sickness and to make it into a comfortable experience for our users. You can see endothelial cells, they are pretty prominent in red, and the user can simply make this data set bigger by pressing and holding their hands and moving their hands apart. Another thing that we implemented here is called anchor control, where you can grab the tissue slice in 3D and then you can press a button and then you can pull it closer. It's kind of like if you have a wand in Harry Potter and you try to pull something closer to you. That's basically what that feels like. And then another thing we implemented is brush and link, where you can brush over these legend entries and then the corresponding cells get highlighted. And that shows you then that some of these cells are all across the tissue, while others, like these keratinocytes here in a second, are more arranged in layers here towards the surface of the skin. Okay, down another floor. This is data from Yang Miao from John Hickey's team at Duke University. And this is currently built with the help of my research assistant, Yash, shown here. This is a visualization of cells from the large intestine. And it is each cell here is visualized 4 times at different aggregation levels. At the tissue unit level, the community level, the neighborhood level, and the cell type level. And you can see as we move from tissue unit to cell type, we get more and more colors as the classifications become broader and broader. And so this is a prototype visualization that we first prototyped in 2D, but then expanded into VR. And we give the user the search cone ability that allows them to highlight the same cell on all 4 layers with these different colors. One thing that we're currently adding for this, and this is a very prototypical shot. I just got this today. Is very exciting. This shows you counts, like how many cells are we seeing, and how many different tissue units, how many different um, communities, how many different neighborhoods, and how many different cell types are actually visualized and captured right now. Again, this is construction. This is all work in progress. And then we're currently working with Alex Wong from Peter Sorgel's team at Harvard Medical School, and we're doing this with the help of my research assistant Keyur. Um, Alex shared with us a series of 3D cells. In this case, what I'm showing you here are skin cells, but we have other cells as well. And you can see these are actual three-dimensional objects coming out of a confocal microscopy, and these cells have very different amounts of complexity. This, for example, is one of the cells. This is a relatively small one with 196 polygons. Polygons, for those of you who don't know, are ways for us. These are basically these surfaces here are polygons. And we use those to measure the complexity of 3D objects. And we also measure it to understand how fluid is this going to run with the VR headset when you deploy it at the end. We have a budget that we can use of polygons in the scene, and we have to make sure to stay under that budget to make sure that the application runs smoothly for everyone. And this is a relatively small one. This is a medium-sized cell with 400 polygons, and this is a large one with almost 800 polygons. So you can see how the topography changes between those. And this is some of this is how we build and prototype these scenes. So, here we take some biomarker values that we have for these cells, and we make visualizations. The basics of what we do in our team here is we're actually a data visualization team. So we try to use data visualizations to find interesting patterns in the data that we could transfer into VR. And here are some three-dimensional prototypes that we're trying to build with what this should look like. And then we're going to go down 3 more levels to the lowest level data that we currently have. This is some storm microscopy, again, given to us by Chloe Lindbergh, shown up here. And this is very much down at a very low level. Each of these are photo switchable fluorophores. This is what you see here, and they all go around a specific centroid because they switch on and off over time. So this is an interesting case because Chloe approached us because she has a headset and she has her own data, and she would like to show this at conferences with her headset. So we built this level for her. We took her data, built this level for her, and then added it to our elevator directory. And now she can go to this whenever she opens up the HRA organ gallery. So here's another comprehensive gallery of the data that I was showing you at all of these different scales and levels. And one thing that I want to hint at now that we're currently building, and this was built by my intern Hunter here, shown on the top right, are multi-scale elevator transition scenes. So we already have the metaphor of an elevator, but we wanted to take this a step further and not just transport the user from one floor to the other. We also want to add an experience of seeing these scale changes in real time. So we always take inspiration from cinema and from video games and from the arts, because part of what informs our work in VR is exactly this, to make it aesthetically pleasing as well. And so this is just very hot off the presses. This is one of these elevator transition scenes where we actually are in a 3D glass elevator, and we are now shrunk down to a much smaller level. And this is then how big the human actually appears. You can see this is the male with the mouth 3D reference object inside of them. And you can also see the lung down here coming out of the fog. And then we have this 2-dimensional graph here that shows where you are currently at. And again, this involves a lot of fine tweaking. We have to make sure that this is not too overwhelming for the user. But these are the ways for us to communicate scale here in VR. Okay, I want to now loop back around to what I said at the very beginning when I was talking about these IRVEs, these information retrieval environments. In 2003, Baumann and al. Showed us this screenshot of a palatine tonsil with an overlay that showed the area for it. And at the same time, they also showed us these semantic objects, which are just, geometric primitives that have these associated abstract related data information pieces. Now, 22 years later, we're in 2025, and now this is what the tonsils look like now. So now we have very high resolution models for these tonsils, and we have all of these tissue blocks inside of these 3D models that are all associated with metadata for the donor that it came from, the data sets that are associated with it, the cell type populations that are associated with it. And so I can't help but feel like we were able to take this concept, which was, in my opinion, so ahead of its time, and implement it now with all of this really high quality bioinformatical data coming out of HuBMAP and ZenNet and all of these other CFDE related efforts as well. And one of the really beautiful things about this is that I was at a meeting recently at Harvard Medical School about visualization and user experience on spatial biology. And by happenstance, I met one of the co-authors of the Bauman paper, and I was able to show her this organ gallery. And this was just a very important loop. It felt like closing, being able to show Jian Chen here, who was this co-author, this application here. So I think, yeah, this paper was ahead of its time, and we're still building on that, I think. So with that, I have my elevator pitch, and pardon for the bad pun. If you have spatial data and you want to see your spatial data mapped and represented in virtual reality, please contact me, and then we can find a floor for you and we can build a new scene for you. And then I want to give a slew of acknowledgements. We have a gigantic team here involved in HuBMAP, working with our PI, Kadi Berner. We also recently got the ninth release of the Human Reference Atlas out. This is the picture that we took a couple of weeks ago here at Indiana University with a few members of the team. I, of course, also want to acknowledge my excellent team of research assistants, interns, and also Ushma Patel, who is the medical illustrator, helping us make the organ gallery into something that you not just can use but want to use. Here are some scientific references. I want to again give a huge shout out to the people that helped me organize the workshop at NIAD, the Powers of 10 workshop, and also the JumpStart Fellowship and the JumpStart Award I received before then that made all of this work possible. All of the people that contribute to the HRA. And with that, thank you very much. And I'm open for questions now.
Hi, everyone. Just a reminder, if you have any questions, please feel free to raise your hand. I can take you off of mute. You can use the chat, or you can use the question and answer. We are monitoring all three.
Out of curiosity, before anyone has a question, I'm wondering how many people have used VR before among the attendees.
I have.
All right. And then let's see. Can the attendees like raise their hand or something?
Yeah, they should be able to, I think, or maybe not.
Okay,
Oh,
no, they can.
yeah, they can.
Yeah, I would not be surprised if many people have never used VR before. If that is the case, I will be attending the next CFDE meeting and I'll have my headset with me and I'm happy to give a demo to anyone who's interested.
That's awesome. Julie?
I guess, Andy, related question. This was a fantastic talk. I actually used VR for the first time when you gave me your demo at one of our CFDE meetings last year, I think it was. So I think this is a really promising area. I guess as someone who's new to it, I'm curious where else you see VR becoming used. And say if somebody invests in a VR headset, where are other places that they could use this type of technology in their research?
That's an excellent point. In so in general, I would say VR is a great vehicle for anything that is spatial. So that can be spatial data, like spatial biology data that's already two-dimensional, three-dimensional. That's an excellent use case for it. It's also very valuable for teaching anything that is three-dimensional. Anatomy is a really good use case. Pathology could be a really good use case. And that's probably the biggest one, outreach and training. One of the really nice things about VR headsets is that they still have enough of a novelty that people want to try it out. So the cost of having somebody come to you and you can show them your own data or somebody else's data or something that you're working on it's just much lower because people are naturally curious and drawn to what you can show them. It's also naturally more immersive. And so it allows you to show things all in one continuous space that you might otherwise not be able to. There is always an interaction cost with VR. It always takes people time to like put it on and get their hair right, and all of these things take time. But if you jump over that hurdle, you can show your own data in a way that you otherwise wouldn't be able to. A fourth item that's really important is anything that involves different scales. If you are in VR and you look at data at different scales, the nice thing is that you always maintain your own body kind of as a reference. And so it really helps you understand how big these scale differences actually are. And so I would say there's quite a wide range of elements where a VR headset could come in handy. I will also emphasize the VR hardware that we use, in the grand scheme of things, if we talk about biomedical equipment, it's a rounding error. A Meta Quest 3, for example, costs I think about $550, which this is not cheap. But if you think about how expensive much equipment that we normally use is, it's a fairly affordable one, and these are really well supported. And there's other headsets too. We just happen to use the Meta Quest platform because that's by far the most widely sold. One of the last stats I read was that Meta Quest 2 and 3 were sold there's more units sold of those than Xbox Series X's, which is insane to me. But
Yeah, that's incredible.
yeah, so that means that there's a market out there for people that can view your applications. And I think it's probably going to go up over time.
Do you happen to know if a lot of labs and I guess schools, thinking about colleges, anywhere that you might have that training, is this something that's becoming more regular practice, or is this something that we're still progressing towards? I'm just curious from that side of things.
I think we're still progressing towards it. Many universities have virtual reality labs or mixed reality labs. They have different names. They all sound really futuristic, but it usually means they have a series of headsets, and then they also have computers to actually develop hardware, to develop software for these. Usually, like at Indiana University, for example, we have one of those labs in informatics, but we also have a much bigger one in the media school. We have a game design program here. And there they have really nicely spec'd out reality lab. So those do exist. And then some universities also have advanced visualization labs that help, out with it. They help with scientific visualization. They provide visualization expertise, but they're a professional service inside of the university. And those usually also have these kinds of headsets. Of course, the cost for these has come down, and they're also much easier to use now than they were maybe 6 or 7 years ago. So more and more, I would say the the action items also shift towards the labs and just getting these on their own. And because they're as easy to set up as a game console, you unpack them, you charge them, they download some firmware updates, and then it's like getting a new phone, essentially. So the hurdle really becomes smaller and smaller.
Thank you. Any other questions from participants? Again, please feel free to raise your hand, use the chat. Oh, I see
Yep.
a message from Doug Keel. Can you show
Yes.
us the list of organs currently mapped?
Absolutely. And for that, I'm going to share my screen again. And I'm going to go to our 3D reference object library. Okay, let me start sharing again. All right, so we are now in the Human Reference Atlas portal, and I'm going to use this opportunity to talk about that real quick as well. This is the brand new HRA portal. We released that just about 2 weeks ago. I think you're the first ones where we actually show this like really publicly. So this is the HRA portal, and on here we have a data section where you can go to 3D reference object library. I want to put that into the chat as well. And in here, if you scroll down, there's a list of all of the organs that we have currently available. And you can either select one of them individually and then inspect them, or you can load all of them at the same time. That's what's happening right now. For now, yeah, here we go. So the male is fully loaded. So these are all of the reference organs that we have for the male body. You can zoom in a little bit here. Can see all of them nicely in here. Let's maybe focus on just one. We have 73 total, by the way, like male, female, left and right. Let's maybe look at the lymph node because that's one of my favorite ones. The lymph node has some amazingly detailed vasculature inside of it as well. And then from here, you can download you can download the crosswalk that tells you which parts of the 3D model are associated with which ontology term. But you can also download the 3D model itself. And then we have summary statistics down here as well. So it shows you for male and female for these individual organs, how many anatomical structures have been modeled inside of there. And these models are free to use. It's a CC BY license, so you can use this for basically any other application as well.
Andy, there's another question from Christy Cano. Is there any way for users to upload or overlay their own data into these models?
Yes, absolutely. So there's two major ways. So one is you can use the registration user interface, and let me just bring this up real quick as well. Let's see. Here we go. This is the registration user interface I was mentioning earlier. And in these models, I'm gonna just put my name in here, and then I can show you. Let's use, for example, let's use the female left kidney. That was our example earlier. Now we are inside of this user interface. We have the kidney here, and here I have a block that represents a user's own data. And this is what you can use to indicate the spatial origin of where the tissue comes from. Now, if you want to make a visualization more like these lower level visualizations, like where you can actually see individual cells, that is absolutely possible as well. That's the second way. And for that, all you have to do is get in contact with us. And then we are building this scene for you. And then we can also make sure that, we can do some quality assurance, even if you have a headset or not. We're working with people that have headsets. We're working with people that don't have headsets. And there's always a way to communicate what the 3D layout looks like. So yes, there's two ways for people to upload and overlay their own data into the models.
Great. Any other questions from attendees?
All right. Well, thanks everyone. Those were excellent questions. Again, if you are at the next CFDE meeting, please reach out to me. I'm usually easy to find, and I will definitely have a station somewhere with maybe our headset in tow and happy to show the application to anyone. I will also briefly put my email address into the chat and my website I put in there earlier. So if you have any questions, please reach out.
Thank you again for the wonderful presentation and for joining us today. And thank you for everyone who participated. As a reminder, this video is recorded and will be uploaded to our YouTube. And our next session will be held on July 25th from 1 to 2 p.m. again. And we look forward to seeing you all there. So thank you again, and we greatly appreciate you joining us, Andy.
Thank you so much for having me. Have a nice weekend.
You too. Bye.
Bye bye.